With this video, we will be successfully done with 2018 question paper and 2019 question paper. Hello everyone, this is Sana Parveen here and welcome to my YouTube channel Sana's Biochemistry. This is our day 12 video of a 45 days NEET practice session. Let us start with question number 1. Which of the following has proved helpful in preserving pollen as fossils? Okay, and the option given to us are pollen peat, sporopollenin, oil content and cellulosic intine. Okay, so here first of all we need to look at the structure of pollen. First of all we will understand what is pollen peat. Okay, now if we have got a pollen grain and this pollen grain has a sticky substance that is present around it. The pollen grain has got a sticky substance present around it, and this sticky substance that is present around the pollen grain is called as pollen peat. Pollen peat is also sometimes called as pollen coat. Okay, and this sticky substance is helpful during the process of pollination, because we all know that the stigma is sticky and the pollen grains needs to go and stick to the stigma. This sticky substance, that is pollen peat, helps in pollination. The second option here is sporopollenin. Okay, for this again we need to see the structure of pollen grain. So we have got this inner layer that is called as intine. Then we have got the nucleus inside, and we have got an outer layer that is present again in the pollen grain, and this outer layer is called as exine. Okay, so we have got an intine, and this green color thing is called as the exine. Exine is the outer portion and it is made up of a very tough material called as sporopollenin. This sporopollenin is actually a biological polymer, and this polymer is chemically inert. The substance that is chemically inert, that is sporopollenin, is not affected by enzymes, it is not affected by acids, neither it is affected by bases. So all these substances cannot do any harm to the exine, that is the sporopollenin. As it is the toughest substance and chemically inert, sporopollenin proves helpful in preserving the pollen grain. The next option, oil content, has got nothing to do with pollen preservation, and the last one is cellulosic intine. This the inner layer, which we are calling it to be intine, this inner layer is made up of a substance called as cellulose. So this is the fourth option, cellulosic intine, but our correct option is sporopollenin, and this sporopollenin proves helpful for preserving pollen as fossils. Next question, which of the following pairs is wrongly matched? Option given to us are starch synthesis in P. Okay, so the first one, starch synthesis in P, and the option given to us is multiple allele. Second is Th. Morgan, linkage. Third one is exotype, sex determination, grasshopper, and ABO blood group system, that is codominance. Okay, let us just start discussing from the option number A. Option number 1 says that starch synthesis in P is linked to multiple alleles, but actually in starch synthesis we have got one gene and we have got two alleles. Okay, as usual, two alleles. When I write the two alleles, it is capital B and a small b. Okay, so I'm going to go for a combination of capital B and small b. This capital B, when it is present, it produces round seeds and long chain of starch. In the recessive one, the seeds will turn out to be wrinkled. It's going to be wrinkled seeds, and the chain out here of starch is short chains. Okay, that means one gene and two alleles. There are only two alleles. Multiple allele case is more than two alleles. But in the starch, we have seen that in the starch synthesis, we have seen it is only two allele. So definitely it is not multiple allele case, and then this option is wrongly matched. Let us just look at the option number 2 and try to get more information out of it. Okay, so the second one here, people, is called as H.T. Morgan and linkage. Yes, it is true. What is linkage? During inheritance, what happens is some genes are inherited together. Why is that happening? Because they are present on the same chromosomes and they're present close enough to one another. Okay, so when the genes are inherited, they're inherited together. That means they're linked up. Which is called as linkage. And this linkage was given by Thomas Hunt Morgan. Morgan is very famous, and Morgan bagged Nobel Prize for his work related to linkage concept. Okay, so Thomas Hunt Morgan, he used to work on Drosophila. That is nothing but the fruit flies. Drosophila, and the linkage concept was given, and that is what the meaning of linkage is, that genes, when they stay together during inheritance through generations without changes, they are inherited or separated as they are present on the same chromosome. The next one is the grasshopper case. Okay, and we need to talk about XO or X0 type of sex determination, whatever you want to pronounce it. Okay, so here, fine. XO or X0 type of sex determination. When we talk about grasshoppers, people, grasshopper, when I go for male and female, the male has got an XO type of arrangement, and the female has got an XX. All the males and the females, they have got autosomes, that is 22 number of chromosomes. But in case of males, we have got only 23 chromosomes because it's an X and O. And in case of females, we have got 24 number of chromosomes. In this, as the male is heterozygous, the male will decide the sex of the coming generation, the sex of the offspring. So this is very true. Grasshopper do follow XO type of sex determination. And the last one is ABO blood group system, which is codominance, which is again very true. And codominance, that means if some person has got an AB blood group system, that means A is also expressed, B is also expressed. A is not dominant over B, neither is B dominant over A. That is the concept of codominance. But when you talk about Mendel, whatever experiment Mendel was doing, it was dependent upon pure dominance. So this concept of codominance is somewhere out of the discussion of Mendel. So all the answers are correct except for the one that is wrongly matched, that is starch and multiple alleles. Okay, let us move on to the next question now. Select the correct match, and then I'm going to read out. Okay. Elect Jeffrey, Streptococcus pneumoniae, option number 1. Francois, Jacob and Monod, Lacoperon, Matthew, Measles and Stahl, Pisum sativum, Alfred Hershey and Martha Chase, TMV. We need to select the correct option. Okay. So let us just start with option number 1, that is Streptococcus pneumoniae. The experiment on Streptococcus pneumoniae was done by Frederick Griffith. Okay, it was Griffith experiment of transformation, that the genetic material can be transferred from one cell to another cell, transformation, and Griffith was the one who set the stage for finding out that DNA is the genetic material. Next one is Lacoperon, that is Jacob and Monod. People, whenever you hear of Jacob and Monod, even if you are sleeping, you should be answering that Lacoperon. Sunato, you should always remember Jacob and Monod. Okay, now what is this Lacoperon, Jacob and Monod? Jacob and Monod originated with this idea that the control of enzyme levels, that means the amount of enzyme level in a cell, is directly proportional to the amount of transcription happening. So more the transcription, more the enzyme will be produced. Okay, and this is somewhat related to the lactose metabolism in the cells. So lacoperon, Jacob and Monod, this is absolutely true. Next one is mesalcil Stahl and Pisum sativum. Pisum sativum, even a 10th standard student hears Pisum sativum and the answer comes out to be Mendel, father of genetics. And what is Pisum sativum? Peas, because Mendel did his all the experiments on peas. Mesalcil and Stahl, they did an experiment in which they found out that they used E. coli. Mesalcil and Stahl for their experiment used E. coli, and then they started to label the DNA of bacteria. Okay, they took the DNA of the bacteria, and the DNA of the bacteria was labeled with heavy isotope of nitrogen. Because they wanted to give the concept of semi-conservative mode of replication. Semi-conservative mode of replication of DNA was given by mesalcil and Stahl, replication of DNA. Okay, and then the last one, Hershey and Martha Chase. Okay, the cursor, Hershey and Martha Chase. Here in this one, they did an experiment on bacteriophage or bacteriophage, whatever you want to pronounce it. Bacteriophage is nothing but the viruses that affects bacteria, that infects bacteria. So if this is the bacterial cell, and then the virus is here, Hershey and Chase showed that the virus is going to inject inside the genetic material and the protein coat stays inside. All these experiments, explanation is going to take time, so we're just going to go for looking of the correct answer out here. So Hershey and Chase, they serve to prove that DNA is the hereditary material. Okay, fine. And TMV is nothing but tobacco mosaic virus. So here the correct option, correctly matched pair is Jacob and Monod, that is lacoperon. Next question. Yes. Which of the following flowers only once in a lifetime? And the option given to us are bamboo species, papaya, mango and jackfruit. It's a direct question. Bamboo species will flower once only in a lifetime. Next question. Select the correct statement. Franklin Stahl coined the term linkage. Transduction was discovered by S. Altman. Spliceosomes take part in translation. Punnett square was discovered by a British scientist. So here the option number 1, which is there, that Franklin Stahl coined the term linkage, is not proper, because we just saw that linkage was given by T.H. Morgan, and T.H. Morgan bagged a Nobel Prize for his work related to linkage. Okay, so here T.H. Morgan. Transduction was introduced by linkage, which just had an explanation. You can just go back in the same video, and then from there you can find out about the linkage. We just had a discussion about that. Transduction was discovered by S. Altman, okay, which is not true. Transduction was discovered and given by Zinder and Lederberg. Okay, these two scientists gave the phenomenon of transduction. Now, what is transduction? In transduction, foreign DNA is introduced into a cell by a virus or viral vector. So here we are using virus or viral vectors for introduction of the foreign gene into a cell. That is called as transduction. And the last one is spliceosome. Spliceosome takes part in translation. Okay, so here translation. If you people know the central dogma is not the mother of a dog. It is the central principle. Every time I tell my students about dogma, they feel that it's dogma, but it is not that way. It is central principle. And what is the central principle? DNA to RNA, RNA to protein. So when you go from RNA to protein, this process is called as translation. Transcription, we need to talk about transcription, that is from DNA to RNA, and DNA to DNA is called as replication. We need to talk about spliceosome, people. Here, for this I'm going to change the color. Okay, yeah, perfect. Spliceosome, I'm going to write it here. Spliceosomes. Okay, So spliceosomes, these are large, complex. These are large, complex molecular machine. Handwriting is not that good, but please consider it. It is, kind of, you can read it. Next one is, it is composed of small nuclear RNA, and it also has 80% of proteins in it. 80% of protein. If you like the video, hit the like button. Haven't subscribed to the channel yet, please do subscribe. And the spliceosomes remove the introns. So for example, I have got a structure out here. In this structure, we have got the introns and we have got the exons. We have got the introns, we have got the exons. Okay, fine. So these introns, we don't need in mRNA, messenger RNA, we don't need the introns. So spliceosome is going to come here and will chop off the unwanted area or the unwanted introns from the structure, and now the mRNA that we are going to be having is going to be all functional, and this is going to have only axon part into it. So that is the function of spliceosome, and spliceosome is found in the nucleus. Okay, and the last one, Punnett square. That means definitely it is not present in translation process. It is present in transcription process. Okay, so transcription. Here I'm going to write. And the last one here, Punnett square was given by Reginald Punnett. So I'm going to write it here. Okay, Reginald Punnett. And Reginald Punnett was definitely a British scientist, and everyone might have used the Punnett square for finding out the ratios of phenotype and genotype. Phenotypic ratio and genotypic ratio. Fine. Let us just move on to the... Okay, so the correct answer here is option number D. Okay, so here we have option number D. Punnett square was developed by British scientists. This is the correct statement. Next one. Offsets are produced by meiotic division, parthenogenesis, parthenocarpy, mitotic division. Okay, people. So reproduction is divided into, or is of two different types. That is asexual reproduction, and we have got sexual reproduction. In sexual reproduction, the progeny, the offsprings that we get are, not completely identical to the parent generation. That means variations are seen out here because of the mixing up of the DNA and because of the meiosis process happening. And in asexual reproduction, we have got the offsprings which are not exactly the clones. In asexual reproduction, the offsprings are exactly the clones, and these clones are called as offsets. So offsets are produced by, in asexual reproduction, we all know that the division that happens is mitosis. So the answer here is going to be mitotic division. Fine. Okay, so meiosis is not the option. Parthenogenesis we need to select. So parthenogenesis is what out here, people? It is that the embryo is developed without fertilization. So there is no fertilization happening. Embryo is developed without the fertilization with the sperm. There is no involvement of sperm out here. I'm going to select a color which is visible to you people. Yes. And the last one is parthenocarpy. Parthenocarpy is a process in which we have got, okay, so yeah, just give me a second. I'll just select the color. Okay, yeah. Parthenocarpy is, where do I write? Is artificially induced production of fruits, and these fruits are seedless because there is no fertilization, because there was no fertilization that happened. Here the correct option is going to be, people, it is going to be mitotic division. Offsets are produced by mitotic division. Next one, the experimental proof for semi-conservative mode of replication of DNA was first given by, we just had a discussion about this, which is Michelson and Stahl. and these people used heavy isotope of nitrogen, that is N15. With that, they found out the semi-conservative mode of replication of DNA, and the option which are given to us is Wieselzel and Stahl did their experiment on what? On the bacterium. And what was the bacteria? E. coli bacteria. So the answer here is going to be bacterium. Correct answer. Next question. Select the wrong statement. Cell wall is present in members of fungi and plantae. This is absolutely true. Yes, cell wall is present in case of fungi and in case of plantae. Mitochondria are the powerhouse of the cell in all kingdoms except Monera. So Monera, if I talk about in the kingdom Monera, we have got prokaryotes or prokaryotic organism. Prokaryotic organisms are nothing but bacteria, and in bacteria we do not have membrane bound organelle. We do not have membrane bound organelles in the bacterial cell. So we do not have that. So this statement is again very true. Pseudopodia are locomotory and feeding structures in sporozoans. When I talk about sporozoans, they show as a non-motile life cycle. They do not have any locomotory organ that is present in them. Locomotory organ or pseudopodia is present in amoeboids. Okay, they are also called as sarcodines. That means pseudopodia or locomotory organs and feeding structures is not present in sporozoa. This option is going to come out to be wrong. The last one, mushrooms belong to Basidiomycetes. Okay, so in the kingdom fungi, there are different types of organisms, different types of fungus are present, out of which mushroom, which is nothing but agaricus. Okay, so this is the mushroom, and the scientific name for mushroom is agaricus. Agaricus belongs to the family of Basidiomycetes, and these Basidiomycetes, again the question was asked, produces exogenous spores, and spores are meant for reproduction. Spores are meant for reproduction. So our answer is going to be here, people. It is going to be option number C. Pseudopodia, locomotory and feeding structures in sporozoans is a wrong statement. Next, Casparian strips occur in epidermis, endodermis, cortex, pericycle. Okay, so for this we need to look at the diagram out here. So I'm just going to draw the diagram so that we understand it properly. This is the diagram from our NCERT textbook. I'm going to label them all. This one inside. Okay. Yes. The first one is called as epidermis. Epidermis. The second one is called as cortex. The third one is endodermis. And the last one is pericycle. After pericycle, we have got xylem. We all know that the plants are continuously involved in absorbing water. When the plants are absorbing water, the water is getting transported into the body of the plant. Okay, so I'm going to take the blue color, and the water is moving in this direction. Water is moving and moving and moving, and then suddenly there is a waterproof tissue that will obstruct the flow of the water, and this is the obstruction that is present out here. This obstruction, which is present near the endodermis, is called as Casparian strips. These Casparian strips will not let water flow from here. As we told, it is a waterproof tissue. Then the water that was moving. Okay, where do I have? Yes. Then the water that was moving, people from here will have to enter the cell and then again come out. Okay, because there's a blockage out here. When the water movement happens in the space, it is called as apoplast. Apoplast pathway of transport of water. And the next one, when the water moves inside the cell, it is called as symplast. Symplast pathway of movement of the cell. So here Casparian strips are present in, it is endodermis. Next question, question number 10. Which of the following statement is correct? Ovules are not enclosed by ovary walls in gymnosperms. Stems are generally unbranched in both cycas and sidrus. Hostales are gymnosperms. Selaginella is heterosporous while Salvinia is homosporous. Okay, so when I talk about gymnosperms, people, gymnosperms, they bear naked seeds. They bear naked seeds. That means ovules are not enclosed by ovary wall. It was a simple question, 4 marks. It is going to fetch you people 4 marks. Stems are usually unbranched in both cycas and sidrus. Okay, so if I try to draw the cycas plant has got a short shoot. And then we have got the soil on which it is going to stand up, and it has got the leaves. This is the plant of cycas, and it is not having any branches in its stem. Hostales are gymnosperms. Yes, a question from plant physiology. You cannot miss on plant physiology because you will be able to see that many questions are asked from plant physiology. This again, question number 10 is again from plant physiology. Selaginella is heterosporous while Salvinia is homosporous. Next question. Okay, so the answer, people, is option number 1. Which of the following statement is correct? Ovules are not enclosed by ovary wall in gymnosperm. Next question. Perfect. Pneumatophores occur in halophytes, submerged hydrophytes, carnivore plants, free-floating hydrophytes. Okay, so people, when we talk about marshy areas, so in the marshy areas, like we call it daldal in Hindi, trees grow there in that area, especially mangroves. So the trees are growing out there. Okay, yeah. So the trees are growing out there, and then as it is marshy area, and sometimes we have got high salt concentration, the plant can adapt itself for surviving in such high salinity. But now what happens is the roots needs aeration. The root needs aeration, but water, there's a hell lot of water because of which the oxygen is not reaching to the bottom. So what happens is the root itself comes out for exchange of gases, and on these roots we have got lenticels present, and these lenticels are involved in the process of aeration. It will do the exchange of gases. These roots are called as pneumatophores. These root people are here called as pneumatophores, and these pneumatophores are also called as breathing roots. As I told, it is then the high salt concentration. Answer is going to be here option number A, that is halophytes, because halo is always to do with salt. Okay, halogens. Okay, NaCl. Cl is the halogen. Halo is to do with salt. Next one, sweet potato is a modified stem, rhizome, taproot or adventitious root. So when I go for drawing the diagram of the plant out here, okay, so this is the stem, and then we have the leaves. Okay, and then we have the leaves. When I look what's happening underground, the roots are modified and they're spherical like this. And this is nothing but sweet potato. Sweet potato. So here the answer is going to be adventitious roots. Adventitious roots. Sweet potato is a modified root, and that is adventitious root. Next question, secondary xylem and phloem in dicot stems are produced by, it's again a direct question. The answer is going to be here vascular cambium. It's a direct question. And the last one is plant having little or no secondary growth are, okay, so growth is divided into primary growth and secondary growth. In primary growth, the plant length increases, the height of the plant increases, and this in the secondary growth, the girth of the plant increases. We all know that in grasses there is no secondary growth because grasses is nothing but monocots. Okay, so the answer of option number A is going to be our answer. So this was all about the questions for today's video. We have successfully completed 2018 and 2019 question paper. That is, in total, you successfully completed 180 questions. If you haven't watched the videos of the previous videos, I'll give the link in the description so that you can go watch those videos as well. I bet it is going to be beneficial. And I'm going to end the video here. Thank you so much for watching. If you like the content, hit the like button, subscribe to the channel. Don't forget to hit the like button so that you'll be notified every time we upload a video. Thank you so much. Bye.